Welcome, everyone. Thank you for joining us in the first of the sequencing virtual seminar series. These will continue weekly for just about the next 7 weeks. We're going to skip middle of October for the upcoming ABRF NeuralSkid meeting. In terms of the logistics, this is a webinar format, so feel free to introduce yourself using the chat function with your name, your institution, maybe where you're calling from. Use the Q&A to ask questions to our presenters. And then if you have technical issues or other questions, reach out to me in the chat and we'll try to deal with that. In terms of other upcoming events, here's the schedule for the remaining seminars in the series. Each week, same time, and the same link and format. You can register for any or all of the programs at the shorter link that we have on this slide. ABRF's regional chapters, we have 3 meetings coming up in the next couple of weeks, next month or so. The first is our West Coast chapter in Los Angeles on September 21st and 22nd. You can use the QR code here to access more information. And then in October, we've got 2 chapter meetings, the Northeast in Rochester, New York, October 12th to 14th, and the Midwest chapter in Columbus, Ohio, October 17th and 18th. And of course, the ABRF annual meeting is coming up May 7 to 10 in Boston. We'll be opening registration for that by the end of next month. For sponsors, that registration will open in just a couple of weeks. So with that, I'll turn it over to Anoja, who is our moderator, to introduce our speaker, if she wants to do that.
It's really nice to have Sean Levy from ElementBio join us to give this seminar today. I'm so glad Element chose Sean because he knows the struggles of core facilities as he has already in the past run them. So in the interest of time, without taking any more of Sean's time, I'll have Sean go ahead. Sean, you're on mute.
Sorry about that. The slide's showing up all right. Should be a title slide, Elements of Freedom. Is that?
Yes.
Great. Just wanted to make sure. So thanks, Anoja, for the introduction and the opportunity to speak here. Having been a participant and member of ABRF, I guess from a membership perspective, off and on for the better part of 20 years. I think this group in particular is one that I'm always very enthusiastic to speak to and interact with, just for the reasons that Anoja mentioned. I'll give a little bit of a personal introduction in a moment, but I think the unique perspectives and even the unique opportunities that core facilities provide are not only extraordinarily important for commercial companies to keep in mind, but I think can also be exceptionally valuable through the whole product life cycle, from initial concept all the way through execution. And so I look forward to any questions that anybody has. Definitely going to try and cover a lot of ground, but I think, knowing the background of this audience, I think we can move through some of these things pretty quickly, and the intent is to hopefully have a pretty engaging presentation. So I'm going to break down this very high level slide because again, like I said, things will be a little bit dense, but I think it'll hopefully be for the benefit. So about 10% of today's presentation is some of my own personal musings, again, mainly because of this ABRF audience and reflecting on some things. About 40% commercial and 50% science, and covering both element sequencing platforms as well as the element loop technology that I'll mention towards the end. But I did want to essentially give you some clear take-home messages right at the beginning that I'll touch on through the talk, but I thought helpful just to essentially put up some of these conclusions right away. So Element Biosciences, I think at least in the field of genomics, is very uniquely customer focused, and following 4 points that we've come out to the market with very transparent and very consistent pricing. And in fact, probably offered the very, very first reagent pricing guarantee, in the sense that Element has committed that it will not raise reagent pricing for the life of the product when they are released. And that is true for the currently available kit. It will be true for the upcoming kits, and is something that the company stands very strongly behind, to allow science to be predictable, to be able to be budgeted very effectively, and with the recognition that if a product is well developed and mature as it hits the market, those types of pricing surprises should be avoidable. And separately, or secondly to the reagent pricing guarantee and that consistent pricing, which are certainly more on the financial and administrative side, Element's also been very careful to not release any fictional roadmaps or claims that are years away. And I think that, this is really I mentioned this to address probably our most common question. The most common question is, it's a benchtop sequencer now. When is there going to be a 5 billion or a 10 billion or larger output on the machine? And that's something that has been very specifically avoided for very good reason. One is, I think the $100 genome is certainly a laudable goal, but the $100 genome is not very valuable as a single $100 genome. It's valuable when you can do it 10,000 times or 100,000 times or 1,000,000 times very reliably with high quality data. And so I think we have the field in general, I think absolutely has a long way to go to really defining what it means to be a $100 genome when you think of the full end-to-end costs, whether that's how you amortize machines, how you store data, how you analyze data, how you combine data. These are all really critically important questions that many outstanding groups around the world are trying to answer. But to give a pretty direct Element specific response, I don't have a picture of a flow cell, but the Element flow cells are the size of a microscope slide, and they have 2 lanes. So from a real estate perspective, in a brute force perspective, Element, certainly the chemistry could be made into a larger flow cell to do larger numbers of reads, but that's not the intent. That's not the point. And I'll explain why as we go through the talk. And so I would say, Mike Prebity, one of the co-founders, who I think is actually listening in here in the talk, would absolutely agree with me when I say Element was not founded under the principles of taking a brute force approach to solve these problems. It's been to take some very elegant approaches to kind of change the trajectory and direction of these technologies. And that remains a key focus and key commitment. Now, there are some very real successes in those efforts, in that Element has yet to revise specifications to the negative or essentially change anything that we've said we were going to do, and that's a strong commitment by the company, which is we'll begin releasing more and more information about our roadmap. But again, we'll be doing so when we know that the science is solid and when we know that the results will stand on their own and be a benefit to the field. And I think that's our guiding principle, is to be a benefit to the field. What we can demonstrate today is that we can deliver 10 times better sequencing at 1/3 the cost if we put this into a very marketing specific message as we compare ourselves to any of the commercially available or even soon to be released benchtop sequencers. And I'll detail that more later. Improvements in sequencing quality certainly matter in results, and I'll show some of that. And then finally, there's lots of details that are critical. Insert size, the ability to do paired end sequencing, all of that matters to many applications, and that flexibility in sample preparation is paramount. And I'll again address that in some coming slides. But really wanted to at least kind of go through some of the conclusions to orient everyone as to the messages that I'll try to illustrate in the coming slides. But I also wanted to mention, I think this resonates extremely well with the ABRF audience, we all aim to accelerate learning through collaborative discovery. I mean, core facilities are the foundation of so much collaboration among institutions where they exist, and even between core facilities and in efforts and organizations like ABRF. And I think we all have appreciated the complexities of biology, especially as we've entered the genomics era, where you can look at things on a genome scale, and that these mysteries really are only going to be understood in that collective effort. And whether we're looking at healthcare or translational medicine or things that I think everyone gets a very personal attachment to. It's easy to get lost in the mechanisms of biology, but when we take a few steps out, we all have the same goal, and that is improving the quality of our healthcare system, improving the quality of life, and improving a healthy longevity for our family members, our friends, our colleagues, and in our society in general. I think I've had so that's some generic images along the bottom, and I won't go through the different stories at the top, but some of these are just highlights that I've had the privilege of participating in over the years, and in many of these have very strong personal or family connections, not to me directly as the investigator, but certainly to the families that we helped, from the two women in the middle who share two different mutations in lamin A and obviously have very, very different phenotypes. The one on the middle right with the muscular dystrophy, the one on the middle left is a bronze medal hurdleist for the Canadian track team in the Beijing Olympics, and able to be impactful in both of their lives, to the two children flanking each side. One of them a very rare infectious disease case that we helped solve at the University of Alabama Birmingham, and the other on the left-hand side, which was an amazing story of a child out of Vanderbilt University. The punchline being is genomic tools is what enabled all of these things to happen. Oh, and then finally, on the two, flanking the two, is the use of these tools in forensic studies to bring closure to families. So certainly not nearly the same positive outcome from an individual perspective, but nonetheless, an amazing application of the tools. So I realized I didn't really have a good summary of sort of myself, and so I'll take a moment to be a little bit conceited, in the sense that a question I've gotten from a lot of friends and colleagues is, well, why Element? Why did you move there? And I thought I'd just orient this to kind of anyone not familiar with some of the things we've done in the past. Wanted to take this as an opportunity to recognize the path that got me here, and give you some perspectives onto what we're doing now. So I have a very convenient time. I got my PhD, which was in 2000. So it makes calculating how long I've been doing this pretty easy. Makes the math very simple, which is helpful. And I did that in the mouse models for mitochondrial disease with Doug Wallace at Emory University. And the images you see are actually some litter mates where we made a transmitochondrial transgenic mouse. And so those are all litter mates. The little tiny mouse you see on the right-hand side carries a protein synthesis mutation in his mitochondrial DNA. In this case, this was a male mouse. And then we went on to analyze that mouse, which was a horrible graduate student project, because literally every mouse that came out, because of the nature of mitochondrial biology, it was truly an N of one. So an absolutely awful PhD project, but nonetheless one that was extraordinarily formative in terms of appreciating challenges and solving challenges, and allowed me to using microarrays, which you see on the bottom, actually use some microarray tools to analyze what was going on in these mice and a couple other mouse models that were made at the same time. So that led me to Vanderbilt University, where started in the core development efforts, and at Vanderbilt established their first genomics core, which was microarray based, and then grew that into a self-sustaining facility that really contributed substantially to Vanderbilt's efforts to double their extramural funding in the time I was there. As we tried to accelerate that into the sequencing space, had an opportunity to join the Hudson Alpha Institute for Biotechnology, which was a mechanism or a way for me to do a lot of the things that we were able to do at Vanderbilt, but just do them at much larger scale. So founded and developed the Genomic Services Laboratory, and then a few years later, the Clinical Services Laboratory. Clearly, I'm not very creative at naming laboratories, but the GSL and CSL became continue to grow to be large facilities. And then in a very interesting twist, the Genomic Services Laboratory was sold to Discovery Life Sciences in the largest transaction in Hudson Alpha's history. And what that essentially did was it laid a financial foundation that will allow the institution to pursue their goals for the foreseeable future. And so I was very proud to develop the laboratory that ultimately got sold. Spent a short time with Discovery Life Sciences as their CSO, which again, very informative and rewarding. But at the end of that, it gave me an opportunity to reflect back and say, do I want to continue to run cores or develop cores? Essentially, it'd be starting over again if I wanted to do that, seeing as the core lab that I had developed at Hudson Alpha had been sold off. So it was a great time to consider going in a different direction. And what I looked mostly for in that different direction was not so much the technology or the team, not so much the technology, but more the team. And that's what I found at Element Biosciences, an amazing commitment to a foundationally new technology. And so I've joined them as the SVP for Applications and Scientific Affairs, and that was earlier this year. And so I'll share with that. And I think the punchline of all of this is the lessons learned for 20 years of being able to work with so many great investigators over time. I think it gave a very unique perspective across a huge diversity of projects, a huge diversity of science, and a huge diversity of challenges, which I think has provided me a background to now approach some new challenges with a new organization with a very unique perspective. So, with that, I think anyone who's seen me speak before, I also use the timeline of my kids growing up as the evolutionary timeline to sequencing, and you're seeing some of those technologies here. This is just to illustrate that, things change very quickly. The only change really is the only constant. And whether it's my oldest son, who was, born in Tennessee, grew up in Alabama, as you can see from that lower left-hand picture, all the way through to just before leaving the Alabama-Tennessee area on the far right-hand side of the slide, with a huge diversity of sequencing, whether it be the ion torrent machines in the middle, to the growing maturity of the Illumina platforms, to Oxford Nanopore, to Sequel II. It all summarizes down to this, essentially the evolution from 2009 to 2020 of going from small numbers of machines to large number of machines, from adding benchtop, adding long read, et cetera, and this opportunity to really walk through this genomics adventure literally over the last 20 years. But interesting things, it really, but if you look back at this, it wasn't that long ago that sequencing Phi-X, like on that foundational Solexa instrument, was, it was in 2005, right? It's, call it 15 years ago, 17 years ago. So now we routinely do human genomes. And so again, there's, a picture of my son, that's him now. He's a freshman in high school, and continues to pursue hockey at a high level, and which has been a tremendous amount of fun to watch and participate in. So if I, now very firmly turn a corner to Element and give you some of the brief company facts for the Element. It was founded in 2017, so it's just at its 5-year anniversary, headquartered in San Diego, with operations in the Bay Area of California. The building you see on the upper right-hand corner is the new Element building. It's about 200,000 square feet of space dedicated to Element, and that includes all of the R&D, administrative, and even manufacturing facilities. So everything we manufacture, and I'll mention this later, is done here in San Diego, as well as in the United States. We're a little over 300 employees. We've been shipping instruments since May of this year. And then you can see on some of the accolades for the company on the middle right, as well as some of the founding investors of the company shown along the bottom of the slide. So a very blue chip set of investors, stable funding base, and something that the investors and the team behind the company has been extraordinarily efficient with their capital and extraordinarily efficient with their technology development. So I'll recognize the entire Element team. Everything you're about to see has been a huge collective effort. And I'll try to call out different groups, but this was a recent company photo that was taken at the San Diego campus. And so certainly recognizing the huge amount of contributions that all of these individuals have made over the last 5 years. And with me having joined the company just a few short months ago, I am truly representing the work of the individuals you see here, rather than the work of myself or a team that I developed. So to give you the quick summary of the instrument, and then I'll show you the data that comes off of the instrument. These are probably the most important metrics. It's a dual-sided instrument, so 2 flow cells. There, one goes in the A, or the reagents go on the A and the B side. The 2 flow cells kind of actually get loaded right above where it says AVITI in the middle, and that dark side or the black middle of the instrument. And then on the very upper middle of the instrument is the touchscreen control. And so it's a fully self-contained instrument. There's no external computer. There's no other requirement other than a network connection to have the instrument function. Does all of its onboard processing is done with onboard compute, and then obviously secondary and tertiary analysis are done offboard on external compute. So 90%, the Q30 specification is greater than 90% for PCR plus libraries. It gets a bit better with PCR free. I'll show a little bit of that. Current kit is a 2x150 read length kit, or a 300 cycle kit. There's an additional 32 cycles available in the kit for use in a variety of indexing conditions. So it's a 4-read system. So you can do, like Illumina has established as a routine workflow, you can do a read 1 and a paired read 2 plus 2 index reads that can be used in a flexible format. So 10x libraries or other things that require unbalanced read lengths are all perfectly well supported. It's about a 44-hour turnaround time on most of the average runs. We put 48 hours as a specification, only because depending on network speed and a couple other variables, the run can be a bit longer than that, but it's a bit under 2 days. And that's for one or two full flow cells at the full output of the instrument. There are some levers that you can pull to decrease those run times that I'll mention in a moment, but generally speaking, think of it as a 2-day turn. Because the two flow cells are fully independent, depending on batching needs, you can start one day and then the next day start the other flow cell, where you actually get a more routine data turn off the machine, where you can kind of essentially have having finished samples come off the machine every 24 hours by leveraging the two independent flow cells rather than having them run simultaneously. So that's certainly an option. And then we spent a lot of time on the compatibility of the library prep. So putting this in context to some of the competitors and highlighting some of the different opportunities and pricing is shown here. Happy to share these tables in case anyone would like to see these in more detail. But bottom line is, it's basically a $5 to $7 per G list price. I mentioned the 2 by 150 read length is a very typical run length of the commercially available kit. We're releasing a 2 by 75 kit later this year. And that, when optimized, that'll allow sequencing read costs at right around 80 cents per million read for counting applications. So pretty effective price point overall. And then the CapEx price of $289,000. So that's summarized in a little bit more clearly here. And let me just get this out of the way. We do have some leasing options, the configurability. I mentioned the dual sides, and then you can see the per kit price. And I think this is, and the reason I left this slide in is to make an important point. And there's a graph that I think illustrates this in a moment as well. But I think there's a tremendous amount of power in an instrument that still delivers a competitive cost per G without having to generate a very large number of gigabases of sequence if they're not needed for development applications, for lower numbers of samples, for smaller batch sizes, et cetera. So you can do pilot work or turn on the machine and run the machine effectively for less than $2,000 a run. So it's $1,680 for the sequencing kit for one side. Obviously, it'd be double that if you run both sides simultaneously. The released pricing for the paired 75 kit is $1,080, and then you have the approximate outputs at specification, at our standard specification, which again, I'll mention some of those details in a moment. 240G for the 1680 or 120G for the 1080. But operationally, most certainly, anyone that's accustomed to running instruments in a core facility where you're routinely running them at their maximum output and at their most ideal conditions, you'll actually see a substantially higher output than what I mentioned. So you'll see that in a moment. So the ask, right, when this was built, was the best in class data quality, having an affordability and a flexibility in workflow. And really, as everyone in this audience knows very well, the traditional mode of decreasing the cost of sequencing was at an ever increasing cost for an instrument and at an ever increasing cost per run. And so what that means is that to get to those lower costs, it's always been a mechanism of scaling throughput, which is where we see this downward trend of the line in terms of price per gigabase, but it always goes with an upward trend of the output per flow cell. Now, to be fair, for many applications, that output is critical and amazingly enabling, but it isn't necessarily for all applications or for all situations. And therefore that was the target that Element aimed for, and is really one of a unique instrument that sits off that line that you see on the graph. The reason the dot for Element is a little bit bigger is, as you'll see in a moment, depending on how you load the machine, you can have that cost per gigabase and that output per flow cell actually be a little bit variable depending on your applications, which can be user defined. So we spent a lot of time in the early phase. Elements really had kind of 3 phases of the company. The first was the technology development and proving out the platform, which occurred from 2017 really until very early 2022, where the company was very quiet. There was very little data that was released, if any data released, and really no specifications. And then in March of this year, Element did a virtual launch where a number of partnerships, a number of data sets, and a lot of the specifications of the instruments were released. And then we amplified that message and added more information at AGBT this year, just before we began commercial shipments of the instruments. And we've now shipped many instruments throughout the United States, and then we'll move into Europe in the later part of this year and early next year. And then it will become a global company by the early part of 2023. Importantly, coming into the game, call it a decade into genomics, but coming into the game really with the mindset of changing the game from a sequencing perspective, certainly could not ignore all of the innovation, the contributions that were made academically and commercial across the sample preparation, library handling, and data analysis fields. And so we embraced that the opportunity to partner with a wide variety of both companies as well as academic collaborators and core facilities, et cetera. And I highlight some of the commercial ones here where we've either had application notes or press releases or a combination thereof, or even some conference talks. So this isn't an exhaustive list. These are just the ones that we've already publicly announced in the past, but want to just illustrate, and I certainly won't read through it, that it covers a broad breadth of applications. It covers a variety of other areas, including resonating very well in the laboratory developed testing space and the translational space, et cetera. I recognize it says clinical on the slide. I do want to absolutely point out that this is certainly a research use only instrument, but some of the capabilities and features of the instrument have resonated well in laboratories that have used research use only instruments in laboratory developed testing. But did want to mention that there's been no formal announcements around paths towards specifically a clinical application other than through that LDT mechanism. So I should probably revise the slide on the in a subsequent presentation. So how does it all work, right? The next couple slides is really kind of taking this introductory stuff and just explaining how it all works, and then really what the foundational pieces of the technology are. And those foundational pieces can be summarized as Element's surface chemistry, and then it's sequencing chemistry. And this slide focuses on the surface chemistry. So what Element developed early on, or what Element focused on early on in the development, was the ability to create call it a biologically relevant surface chemistry that did not have high background under a wide variety of conditions. And all of those points are quite important. So what you're looking at on the right-hand side of the slide, and I'll pull up a laser pointer here. Here's you're looking at an example of a high background surface chemistry, and then in similar conditions with Element's high CNR, which stands for contrast to noise ratio, with a very flexible ability that you can have an Element substrate with surface reagents, as well as custom nucleic acids, proteins, or other biologically relevant molecules or substances that can be incorporated onto the surface chemistry to perform some downstream assay. And sequencing is the first downstream assay that we're commercially launching. Now, it's not to say that you can't do other things, and these are just some, again, examples that we've released already. No specific applications or data are attached to them at this time, but just illustrating the flexibility of the surface chemistry. And you can see here an antigen, a plus and minus antigen example using some fluorescently labeled antibodies, as well as a cell culture example. You're looking at cells through the same optical machines. Critically important just to mention, the images you're looking at were in fact captured on the same optical system that's in the sequencing platform, giving some flexibility there. Giving some indications of where the future flexibility is on the platform. We're extraordinarily open to ideas and brainstorming with members of the academic and even commercial communities about how these surface chemistries and sequencing chemistries can be applied in unique ways. And so delighted to talk with anybody in the future. So in 2018, the amplification methodology that was used on the flow cells, which is a rolling circle amplification, kind of looked like what you see here on this side of the slide. A little bit of an elongated DNA molecule, not a particularly punctate staining that you could clearly quantitate and do signal or sequencing analysis here. But over the course of some evolution within the company and some additional technology development, they're able to achieve what you see here in the 2020 version, where nice tight clusters and the ability to clearly see 4 different colors simultaneously on the same during the same imaging system. And this is just in this case, this is a PCR-free linear amplification on the low binding surface. So as I mentioned, it's a rolling circle based amplification. This also is a reminder to mention a couple of things that are probably some good housekeeping items for this technical audience. It is a random flow cell surface chemistry, meaning it is not a patterned flow cell. So we do rely on random association of or random generation of these clusters, and then finding those clusters and doing the downstream analysis from there. And it is a 4-color platform. All 4 bases are directly detected and imaged. Where some of these benefits come out can be shown here. The rolling circle nature of the amplification allows for very low optical duplication rate, which is summarized in this graph here. There's another competitor on the market that also uses rolling circle, so you can kind of see that that is one contributing factor to a significant difference between other amplification, whether it be bridge amplification or other methods, which inherently have a higher optical duplication rate. The 4 bars shown under Element are just simply 4 different library types, 2 genomes, 1 RNA-seq, and then 1 E. coli. And then what's shown on the other 2 graphs, and I may have a slide that shows this a bit bigger, but the punchline of these is I mentioned the 800 million paired read specification. Note that this is Q30 here and then Q40 here, with read densities of 900 million paired reads all the way out to 1.3 billion paired reads. So about a 40% change in the output of the instrument. And you can notice what is the effect of the DNA of the sequencing quality on those libraries. So it goes from about 97% Q30 down to about 93% Q30 here. And then same thing, a similar reduction in Q40 score from the high 80s down to about the mid 70s. With that same increase in density or increase in output. So what this means is you have the ability to run the instrument towards specification, towards that $7 per G, keeping data quality as high as possible, say for variant discovery exercises, the highest sensitivity in, say, somatic variant detection, et cetera, or for counting applications where that absolute Q score is not quite as vital, you can push the output of the machine upward by roughly 40%, and still maintain leading Q scores for an instrument, and say whether it be on the Q30 or the Q40 side. I think importantly, and again, I'll just mention this briefly. I won't go into the substantial amount of data that Element has on some of the variability we see. There is absolutely a sample input type, library insert size, distribution of that insert size relationship to all of these things. And that's true with any sequencing platform. So there's a lot of levers that can be pulled and a lot of optimizations that can be made depending on a number of those variables. And I mention them just to also state that we have some of the things we'll provide early access to are some tools for incorporating library metadata into the sequencing setup process to help with loading of the instrument and making sure that expectations at the beginning of the experiment are fully met when you get to the end of the experiment, even when you have some of those variables in there. And I think that's been something that Element has embraced robustly based on customer feedback, based on some of the influence that someone like me who's come up through the core facility ranks have really impressed upon the R&D group as to what's critically important. Essentially, Element doesn't have index hopping. We still, of course, support unique dual indexing, but it's not really a requirement. You can see the very, very low index misassignment rates over 4 different library types. Some of what you see here and the reason for these bars being larger or smaller, but all of them really well below a 0.2% index hopped. So most of this is from either a small amount of image misregistration, which can drive a very, very small percentage of this. And then other parts of this is actually due to index oligo contamination. I'm sure many people are familiar with that. So now I want to mention a couple other data points, but I also want to go through the sequencing chemistry, and then I'll move a little bit faster through the rest of the data slides. And now that once we have this, I think this important introduction to the chemistry, and again, that second of the 2 truly unique foundational pieces of the platform. And that is, so we mentioned the surface chemistry, very, very low background, very, very low binding, essentially no surprises on the surface chemistry when you do amplification, when you essentially set the stage for your sequencing chemistry to do its work. So with that surface chemistry being predictable, very reliable, easy to make at scale, those are all requirements of the flow cells, having learned lessons from, the trials and tribulations of technologies that have come before. Now we shift our attention to the sequencing chemistry. So we'll use one color as an example on the slide, but of course the same theme applies to four different colors. And I meant to make a slide, but I didn't, to just kind of orient the sequencing by synthesis family of technologies versus other technologies that are kind of up and coming. I think Element's the first commercially released technology that departs from sequencing by synthesis for a next generation sequencing platform. So whether it's an Illumina or an Ion Torrent, Singular or even Ultima, a number of different chemistries that rely on the use of fluorescently labeled or other modified nucleotides for direct detection of the incorporated base during a synthesis step, or in the case of Ion Torrent, the detection of a side product of that synthesis step. They all kind of rely, they all have variations on that same theme. You're relying on the incorporation of some modified nucleotide. What Element's done is approach this quite a bit differently, and really puts it in, to be fair, and more of the element chemistry belongs kind of more in the family with Omniome, which is a sequencing by binding chemistry. Those are more related than Element is to the sequencing by synthesis chemistries. So that means you have the ability now to choose different platforms based on their specifications and even based on the bias that may exist in some of the chemistries. So what you see on this left-hand side of the slide is a couple of key innovations that have led to important benefits on the chemistry. So the first, which is here at the top, which has allowed us to be so confident in the reagent cost, prices, structures, as well as guarantees, et cetera. The amount of fluorescent dyes and some of the more expensive reagents that are used on the Element platform is relatively low. In fact, the traditional Illumina-based chemistries or other direct fluorescently modified nucleotides typically have 1 to 10 micromolar of nucleotides in each reaction. Whereas in Element's case, that's been reduced to tens of nanomolar. The clearest evidence of that is if you look, if everyone that's seen the Illumina reagents where you have that deep purple reagent, which is which contains all the fluorescently modified nucleotides with the fluorescent dyes contained in them. It's a beautiful purplish kind of bluish color. It's a very cool reagent to see, but that just gives you an indication of how much dyed material is in there. Where in contrast, Element's same sequencing reagent has a similar overall color. However, it is almost transparent. It is almost clear, which again just illustrates that change in those two things, or change between those things, which really allows significant management of cost of goods, et cetera. So that's not the most critical part. It's an important part, but not the most critical. But what really is critical to appreciate here, and I'm sure everybody's been kind of watching these images, these kind of rotate through the different steps in the chemistry process. Element uses a multivalent avidity-based sequencing chemistry. And so what that essentially means is think of it more like an antibody, think of it more like a multivalent drug, think of it as the ability to sense at multiple locations or multiple bases simultaneously before a measurement is taken. What's shown in the cartoon is just an example of the avidite core, which is this part of the molecule right in the middle, and then it kind of has 4 tentacles that are shown in this cartoon representation, and those 4 tentacles interact with the same base position at 4 different amplified molecules all in the same spot. So again, remember the rolling circle amplification. You make several hundred copies of the same fragment of DNA within those within of course that circular molecule of DNA. You have again multiple copies, and you're sensing at those copies. So what essentially is happening here is Element has split the trapping process, which is this process you see here, from the stepping process, which is when you step the nucleotide forward one base. So that stepping process uses completely natural nucleotides. The only modification they have is a 3' block. There's no fluorescence on that. And that uses a separate enzyme that is used for the trapping and then sensing step for what nucleotide comes next. So by separating trapping and step, you can have optimized enzymes for both. And that was another key area of development for Element, was the protein engineering and evolution of those enzymes for those 2 different activities, each independently performed and therefore independently optimized. Because there is not a fluorescent modification on the nucleotide, it allows the growing nucleotide chain is unscarred. There's no scarring that's left behind due to chemical cleavage or modification, or even the residual remainder of any of the fluorescent dyes that may be left behind it for an incomplete cleavage reaction. So that allows much of the phasing and pre-phasing issues to be masked, or to be not observed in the sequencing chemistry. So, the last thing to mention about the avidity-based chemistry is that in a case that you have a small amount of phasing or pre-phasing error, meaning that a base gets ahead or a base gets behind, because of the multivalent nature of the detection, very often you can still sense those bases very accurately, even when you start building up some of those events. It's also important to mention that if you have, in cases where there's damaged DNA that has been used, where during the rolling circle amplification, some of those either base adducts or other issues have incorporated, or we end up with some small amounts of error in that case, again, that multivalent quality can help balance that and still maintain a high accuracy. So when you take all that together, this, primarily of the splitting of the stepping and trapping and then the unique chemistries, that's what drives the ability to push the Q scores out significantly higher, which is what's shown here on the right-hand part of the slide. This is our observed versus predicted Q plot. And this is for read 1 here and for read 2 here. The predicted is the solid line that you see on the 45 diagonal. The observed for a completely independent library that was not part of the training process is shown in the dotted line. That's shown here again, read 1, read 2. And then the relative frequency is summarized on the right-hand side of each of the plots. And I'll just draw your attention to notice that the vast majority of sequences are in this case, which again, it's a high quality DNA sample into a PCR-free library. So from an element sequencing quality perspective, the ideal substrate to demonstrate this data, to be fair. But we see where, most of this data, greater than 60% is at a Q score of Q44, Q45. And then we see this small tailing off as you go down with really everything above Q35. A little bit of a decrease in read 2, that's routinely observed. And then I'll just mention that, highly uniform ATGC coverage, which you see here at the bottom, the 2 red lines indicate 20% to 80%. So you can see a nice, very flat curve across GC ranges, both in terms of the human genome that we've looked at, as well as some other GC rich or GC poor genomes. What does this all lead to? And I'll get into some other data types on SNPs in just a few moments, but just wanted to mention that our work, as well as the work of many of our collaborators, have shown that we have industry-leading indel calling. Our accuracy and sensitivity of indels is quite a bit better than other platforms or other data sets that have been released on a variety of platforms. The mechanism for that is sequencing chemistry dependent, and it's probably most likely, and this is some of the analysis we've looked into, and we're continuing to look into this, is based on our ability to have an improved context error as you come out of homopolymers. So we get through the homopolymers well, but then as you come out of homopolymers, the element error rate is significantly lower than some of the competing technologies, which appears to be that mechanism. So couple other facts and features about the platform. It's a very large field of view optical capture or optical system that's shown on the left. On the right is just indication that Element spent a long time in developing the algorithms for the spot finding. So because we are using a random flow cell, had to make some improvements there to drive that efficiency and performance. And then a couple of things about the platform. Full remote monitoring is possible on the instrument, as well as very detailed telemetry data for run level metrics and run level performance. Those tools have been developed and they're readily available on the platform. We mentioned the compatibility, the embracing of everything out there for the library preparation that's been driven by the success of the Illumina platform over the last 10 years. So I won't belabor this other than to say there's two streamlined workflows. One is an Adept workflow, which you can take your favorite library type, your favorite workflow, and then at the end at the post pooling step, or essentially when that library would typically go on to, say, an Illumina sequencer, there's an adaptation protocol which does not add any additional PCR or other amplification to it. It essentially circularizes that library, and during that circularization process introduces an element-specific sequence, and then it's that sequence that we use to seed the rolling circle amplification. So you can adapt an existing library onto the platform. We also have element-specific workflows to allow those adapters and primers to be incorporated right away. But again, the punchline being extremely low switching costs in terms of being able to continue to use libraries that you prefer or libraries that you like, And same thing on the FASTQ files on the backside. And you'll see some data analysis where we intentionally kept the bioinformatic pipelines precisely the same when we did some head-to-head comparisons with other data types to again, make sure that the focus was on the sequencing performance and not necessarily differences in the bioinformatics. We see same indexing performance if you take a same library and run it on the two platforms. And just as a note, when I was fortunate to be part of the beta testing program for the Element platform, and we actually used it as a top-up machine for a NovaSeq just as a test, and it works exceptionally well for that. Error models are completely different. The data is completely integratable, and so it is a very efficient top-up machine for without having to go into more more very expensive flow cells, just as a notation. I mentioned we manufacture everything here in the United States in San Diego. That's both instruments and reagents, and we do have now a fully supported or fully capable service and support team across the United States. And then we've just started building out that team outside of the United States as we get ready for the global launch. You saw this in the previous slide. I just want to mention that these data sets are publicly available. This is that PCR free run. If we do the same analysis on a PCR plus run, you see a little bit of a difference here in the top end, bit of a difference here in the divergence. You're starting to pick up errors that are introduced during the amplification process of the library prep, which is why I mentioned, the applications are so critically important. But, when the sequencing quality is high enough, when you get up, into the mid Q30s and above Q40, you really can start seeing, what are the effects of the true end-to-end process in this. So again, on this slide, I mentioned all of these data sets are available, fully transparent in terms of being able to do that. Also released all the information, and we intentionally use publicly available tools for doing our recalibration of Q-scores. So again, anyone can validate all of this data that we're showing around around our ability to get out to those Q-scores into and above Q40. I mentioned this already in terms of the GC coverage. This is just a little bit of a clearer plot. Talked about this as well. In the interest of time, I'll keep moving on. But again, you can tune the machine based on application in terms of you want a bit higher quality with closer to specified output, or you want to push the output of the machine, slightly above or even significantly above that specification, with the knowledge that you'll decrease your Q30 quality or Q40 score by a marginal amount. We validated things like, Olink's platform, 10x single cell. These are all data sets that are available for public review and analysis on our website. But now I wanted to focus on, the whole difference between features and benefits. And I'll kind of skip over this slide in the interest of time. But the idea is the perspective that you look at some of these opportunities, I think, may seem very clear at the beginning, but when you really sit down and think about it, you may revise, your thinking. And this, a great example was from World War II fighter planes, right? They started looking at them and look at all the bullet holes in the planes that come back. We should add armor to where all the bullet holes are, only realizing that, well, the real story was the reason that the bullet holes were in those places. Those were the non-critical areas of the plane as far as actually getting back. And so it was more important to armor the areas where you never saw bullet holes. So, perspective is important as we look at features to benefits. So we talked about the detection chemistry. I talked about the surface chemistry and optics. Obviously, there's diverse biological applications. I showed you some examples like cell analysis or antigen detection that we've done just as, very, very basic flexibility type things. Mentioned cost in terms of doing some of the more bulk type assays. We have a clear mindset towards flexibility, the number of partners, the way we've embraced the data analysis, et cetera. And then, of course, probably the most important thing is, what does the data quality get you? And I'll mention a couple of those things here. So in collaboration with some investigators at UCSD, we had an opportunity to work in some rare eye disease. What I really want to focus here, although this was a really great project to do with that group, just wanted to show that when you push that density up 1.1 billion reads, you can absolutely get about 320, 330 gigabases of data, very, very high Q scores and index assignment rates. And if you add these up, these were intentionally done at 20x coverage for the parents, and then the balance of the coverage, which led to about 52x coverage on the proband. You can do 3 human genomes per flow cell. So you can do trio sequencing on a per flow cell basis with a very high degree of reliability and a very high degree of accuracy, particularly if it's PCR-free libraries that are being put onto the machine. So there are very tangible benefits to pushing the output. If we look at coverage and performance over coverage, this is an example where this was done by Andrew Carroll. If anyone was at AGBT, you've seen some of this data, but it's important to reiterate. Here you're looking at coverage dependency versus total error for SNP and indel false positive and false negative together on HG005. So the publicly available Illumina data is shown here in yellow, and then you have Element run under the Illumina data model, meaning this data went through the exact same informatic pipeline. You can see about a 50% chain decrease in error rate as you go from Illumina to Element. And then by using an element-specific deep variant model, we can decrease that even more by about 6% or so, getting below 100,000 total errors at 15x coverage. You can see things kind of collapse out here in terms of total error at 30x, but that doesn't actually really tell the entire story. We're working very hard to understand this delta, and I'll give you some vignettes into where some of that comes from. So here, if we look at this data a little differently, here's 15x versus Element and Illumina, both at 15x coverage, where the platforms make calls, they're both accurate. This is not, we're not suggesting at all that Illumina sequencing is not an accurate platform. It absolutely is. Everybody knows that. What we're saying instead is that in the noise, that if you can decrease the noise for greater efficiency in the overall platform, and that's what we're looking at here. If you look at the number of false candidates at 15x versus 15x, you see it's significantly lower. This story doesn't change in an overly dramatic way when you go to 30x, although that total error was collapsing when you look at the totality of the variant calls, which again, I will absolutely should note that this is truly a factor of both the sequencing technology, the sequencing chemistry, and the primary, secondary, and tertiary analysis tools. So I'm, yes, telling an important part of the story, but it is not necessarily the complete story, that there is opportunities for continuing to look at how we align data, how we call variants, and how we filter variants. The point here is that the higher quality data coming off the sequencer leads to lower noise in all of that downstream process. And we're very confident that we will be able to continue to demonstrate a higher degree of efficiency as you go through that secondary and tertiary analysis. And you can look forward to some data sets and things coming out on that in the future. This is just to show that, not going to go into this data other than to say, the data that I'm showing you is highly reproducible, highly reliable platform, meaning that these aren't golden runs. This is done again and again and again on multiple instruments. Element has 28 instruments in-house. Those 28 instruments collectively have run almost 15,000 runs. And then we have, of course, the instruments that are now in the field that have been shipping during the summer. Continuing with some of the work from Andrew Carroll at Deep Variant, wanted to focus on de novo calling, right? This is using Deep Trio. And what this is showing here, focusing on the HG00, 3, and 4 trio, chromosome 20, continues with that same theme. In this case, the true de novo calls are shown in blue. So when those calls are made, they are accurate. But note in red the false de novo calls on Illumina compared to Element on the same data set run through the same analysis pipeline. Again, it's that lower noise level leads to that lower false candidate rate that we observe. So that, again, that quality makes a difference. It makes a difference in how many variants have to be validated downstream. We've also looked at low complexity sequencing. I'll breeze through this, and again, in the interest of time, other than to say, we started off like many platforms, and that was the requirement of significant amounts of phi-X to be spiked in a run to do things like low diversity sequencing, say Amplicons, or things like methylated, bisulfite converted libraries, where you kind of have this sort of 3-base genome phenomenon, which makes sequencing quite challenging. So we addressed that, and what I will just call your attention to is here on the right-hand side of the slide, that notice this very similar pass filter densities. This is our pass filter rate, all hovering around 93%, and this is the amount of phi-X that was incorporated into the run. So NA is 0 phi-X, and then we had a 2% or 5% target, and you can see the percent Q30s are pretty similar. As we've done more and more replicates of this, what we do see is as you go from 5% down to 0% phi-X, and if you look at the totality of the run on a per cycle basis, you actually will see a slight improvement in terms of the overall, the shape of that Q30 curve with the 5% versus 0. So we're continuing to recommend that 2 or 5% ballpark for things like methylated methylated DNA analysis through a bisulfite converted or similar, whether it's NEB's enzymatic conversion, which is what was used in this case. Really important possibility though of not losing 20 or 30% of your reads to a phi-X spike. So does this translate to usable data? In this case is going through a Bismarck workflow. Some of the housekeeping things here, 320 million reads. Alignment rate, which hovers in the mid 80% for these, which is pretty typical for those converted libraries. Relatively short insert sizes, and then theoretical versus observed coverage. And then we really wanted to see, using NA12878, did we see the expected 53% CpG methylation? And that's what we saw here. If we compare that to a control, looking at lambda, we of course saw almost 0, and that's what we expected. We're at a little over 1/4%, and we were expecting 0. So it's showing that it was nice and clean. And we've seen this again and again. When we look at a variety of applications, we see that about 10x improvement in sequencing quality. Mean quality scores on Element in the mid 40s. Same libraries run on Illumina, we see those mean quality scores in the mid 30s. And so this is kind of backing up my earlier claim of that 10x improvement. We have some upcoming results on some single cell ATAC-seq and some metagenomics where these same results, continue to drive things forward in a very positive way, that the improvement in the quality, the improvement in the flexibility instrument absolutely makes a difference. And so we very much look forward to getting some of this out. The reason you're not seeing more details in these today, these were part of some of our partnership studies, and we, of course, want to be able to release that information in a complete and detailed manner with the accompanying data sets, so that any of the conclusions and results that we draw can be validated. But these are some of the highlights that you see. Last thing I'll spend time on, and I was hoping to get through some of our loop technology, but I think in the interest of time, we'll have to save that for another day, because I think this is particularly important in a last point. So this is very new data. I apologize that it's not particularly polished, but this gets back to the earlier comment about the importance of the bioinformatic pipelines and the absolute critical nature that as more sequencing technologies come onto the market, which will give an amazing amount of choice, an amazing amount of flexibility to the investigators, to the scientists in the field. We have to continue to take a holistic approach. We have to continue to look at things with a very critical eye and not focus on single metrics and single specifications, whatever they may be. And here's a case in point. So what you're looking at here is using simulated data. So this is if you take use available genome in a bottle type information or reference information and tools like NEAT to make a simulated data set, and then run that simulated data set at different insert sizes, paired end versus single end, different error models, et cetera. You can start asking questions about the bioinformatics pipeline, about what the error models really look like. And this summary, and we're working on a publication on this right now, but I wanted to put out some of the highlights. So this is if we take the NIST V421-2 allele frequencies, put them into a simulated data set. So what this means is we have completely held sequencing error to 0. So this is, again, to be clear, this is not an element specific sequencing demonstration. This is asking a question that if you modulate single end versus paired end, modulate insert size, and then put different numbers of variants into a genome and use our traditional bioinformatic analysis, what do the error modes or error models look like? So on all these plots are oriented the same way. You're looking at total false positive plus false negative errors, and hopefully you can read what's on the screen. What's red is the passing indel false negatives, orange is passing SNP false negative, so you see that is the majority of the error, and then you have passing indel false positives for indels in green, blue is passing SNP false positives, and then the others were the gray is unimportant in terms of this. It's really a small number. Very, very simply, you can just keep an eye on the total error rate. So again, this is zero sequencing error. And this first plot shows, well, what is the effect of insert size of only doing single end sequencing? So single end 200 versus single end 300 versus single end 400 versus single end 500. So with a single end 200 read, you'll generate about 60,000 errors in the analysis, and that'll decrease down to about only 1/3 of that, down to about 21,000 errors by simply having a read length go from 200 to 500. If you start bringing in paired end approaches, 2x150, and then the last one contrast those, but note 2x150 at 35x, these three differing conditions have slightly different insert size distributions, but you can see hovers right around 21,000 errors. If we collapse the insert size down quite a bit, you can artificially push that error rate out. This is due to lack of accuracy in the alignments. And then this very last panel, probably the important point, is if you relate them to each other. So kind of keep all the other conditions the same and take a 1/300 read at 35x versus a 1/300 read here, another replicate at 35x with some slight differences in input material. And then compare that to a 2/150 versus a 2/150 with same thing, slightly changing the statistics of the insert size distribution. So keeping everything else the same, difference between a 1/300 run is about 35,000 total errors, and that drops to about 27,000 errors by simply taking the same number of cycles, 300 cycles, but putting it into a paired end approach and keeping the insert size right around 300 base pairs for the insert size. If you push that insert size out, the error rate drops. So we're working on more and more of this story. So like I said, a little bit unpolished, a little bit preliminary, but the whole punchline of this is the informatics matters. How we look at error really matters. And then certainly insert size versus paired end versus single end, those all play application specific roles. If you're doing counting applications, obviously all of this stuff that I just showed probably doesn't matter much. But if you're doing somatic variant or de novo variant detection, it's absolutely critical. So we have to look at these things in a holistic manner as we evaluate technologies and specifications and capabilities. And we just have to have I think now is an amazing time to be a scientist in genomics, because we're starting to see a huge amount of choice. And I think, on behalf of my colleagues at Element, we welcome the opportunity to walk through, the types of data we're looking at, taking this very science forward approach and hopefully helping people understand of where the Element sequencer can fit into a diversity of applications. We're certainly not suggesting it's a one-size-fits-all instrument, but we are very confident in the impact it will have across a majority of applications. So, like I said, we'll have to save LoopSeq for another day. Let me see what the questions are. Oh, hold on. I may have to.
Yeah, fantastic talk and really cool technology. There are about 5-6 questions on the chat.
Yep, just got to it. So I'll just kind of run through them real quick. How do we see core facilities already in Illumina switching to Element? I think it's going to be a core by core decision. So one simple example is would I being a former core director, would I tell somebody you should replace a NovaSeq with an Element box? No, probably not. But at the same time, can I describe a pathway to have a probably 1 or 2 Element instruments complement a few NovaSeq instruments? 100%. So that's, I think, maybe one type of question. The other is if you're a core facility running NextSeqs, absolutely a much easier commercial sort of argument. We're about half to 1/3 the cost of sequencing on a NovaSeq. And in many cases, you can, if you run a NextSeq, if you run a NextSeq commonly, we can show you the math that you can pay for a NextSeq in roughly 13, or you can pay for an Element machine in roughly 13 months with savings on a NextSeq. The other factor with the 2 flow cells on the Element platform, it's kind of the equivalent of roughly the equivalent of 2 NextSeq 2000. So it's sort of 2 machines for the price of 1. But best answer for that question, it's core facility dependent, but we've tried to keep that switching cost or that incorporation cost of this platform as low as possible. You can use existing workflows, you can use existing bioinformatics. It should be very, very close to plug and play. The next question was, how is the Q-score affected when reading through low diversity regions or working with Amplicon libraries? I mentioned that as part of the methylation data. Happy to share that data and have a more detailed conversation, but it's a minor effect. So we still recommend that 2 to 5% Phi-X primarily so you can monitor Phi-X error rate. So if you really push the extreme of the diversity, you can at least monitor that error rate. But the nature of the sequencing chemistry, once the Polyny locations or the cluster locations are found, diversity can be almost 0 and still work through those regions very, very well. And again, happy to explore that in more details. So does the Adept workflow lead to a loss in cycles that are required to read through the additional length of the third-party adapter? No, so that there is no, you're not reading through any of the adapters. We still use the same indexing and priming sites. So the addition of the element specific sequence has no effect on the sequencing reads. You're not having to read through or get through through any of that. It's completely transparent. We have a mixture of sequencing primers that include the necessary primers to sequence a wide diversity of libraries that can be used. We have about 35 or so validated catalog numbers on our website that describe everything that works completely out of the box. Anything else, we're happy to work with someone on a case-by-case basis. We haven't had anything yet that we haven't been able to adapt, but there is not that element-specific sequence does not affect the actual sequencing process of that native library. Which vendors are you working with for library construction? Are the library preps automated for a streamlined workflow? So yes, there's we've worked with all the major vendors so far. New England Biolabs is both a customer of Element as well as a partner. They'll be releasing some kits specific to the Element workflow. We have application notes with Watchmaker, with Roche, with Twist, with Agilent, with QIAGEN. The list goes on and on. But obviously, we would welcome the opportunity to address those in a more protocol-specific manner. But we've also worked with some of the microfluidic-based automation companies and shown performance there with Volta and Miraculous and a number of others, as well as we have some upcoming collaborations with some other automation vendors to be sure that we can fit that through. At the end of the day, any existing automation workflow that generates an Illumina library can go through an Adept process as well, and we can help with the automation of that. And the last question is, does Element have a program for trading in non-Element sequencers for a discount, for example, in NextSeq? Unfortunately, not at this time. That's just something that we've really committed to that transparent pricing, very consistent pricing, and therefore a trade-in program is not part of that program at the current time. But our commercial team would be delighted to talk with anybody along those lines, but we don't have a formal training program right now. Last couple of questions, plans for single read kits or counting applications. The paired 75 kit will be probably the best for counting applications. Now, the 300 cycle or the 150 cycle kits can be used in a single end fashion. You can just run them at, say you theoretically, you could run a single end 300 read today. You could run a single end 150 with the upcoming 150 kit. And then are there special considerations for base diversity, signal intensity, pooling and indexing? No. Outside of the finding of the Polynese initially, those considerations generally don't apply. But again, happy to, realizing there's a lot of nuance. There can be nuance in that question. So generally speaking, no, but it can be anything else. And then, yeah, that's actually the same question ended up in both places. So Anoja, again, thanks for the opportunity to speak to the group. I think we're probably just a couple of, we are a few minutes over, but thanks for everybody's attention and spending the extra time as we went through the questions.
This was fantastic, Sean. Thanks for getting us started for the series. Have a nice day. Thanks, everybody.
Thank you all.